Hi, my name is Natalie Alvarado. I am a senior in environmental engineering at the University of Colorado Boulder. This summer, I did an REU at Colorado State University in the mechanical engineering department. And this is my project, a sustainability assessment of protein extraction in a microalgae biofuel biorefining concept. So first, let's talk a little bit about global warming. As we know, global warming is impacting our society more and more every day. And one way to help combat global warming is to decrease greenhouse gas emissions. Well, we can see in 2019, transportation accounted for 29% of all greenhouse gas emissions. One way to decrease this percent of emissions is to use biofuels as an alternative to petroleum. The problem with biofuels, specifically microalgae biofuels, is that it's pretty expensive and it's not at that cost competitive level with petroleum. So in order to get it up to that level, we need to use co-products such as protein. So I came up with a protein extraction process, which is down below in the materials and methods section. So the algae first goes into this bead mill, where basically the algae is hit by a bunch of beads, which break apart the cell wall easier. And then the algae goes through a centrifuge, which separates solids from liquids, and then into an enzymatic hydrolysis, where the enzyme lipase breaks apart the peptide bonds to get the amino acid chains, and then into another centrifuge to separate more solids from liquids, and then into a tray dryer. So we have this outputted dry protein algae that we can then sell for bioplastics, animal feed, and much more. So I did a life cycle assessment on this entire process. And as you can see from this first chart, Tracy impact results by process. So we looked at 10 Tracy impact categories, which are environmental impact categories that kind of show how much damage each sub-process is doing to the environment. And as we can see, that orange part, which is the enzymatic hydrolysis sub-process, by far has the greatest amount towards each impact, which means it's the worst towards the environment. So then if we think, okay, why is that process so bad for the environment? Then if we look at the global warming potential of process inputs chart, we can see that out of all of the inputs, that tris HCl buffer has the greatest global warming potential, which means it contributes the most CO2 equivalent in the environment across all of the other inputs. So that input is actually going into the enzymatic hydrolysis step, and that's why the enzymatic hydrolysis process has all of these negative environmental impacts. So then I did a sensitivity analysis and basically said, okay, let's look at these inputs that are having the worst effect in our environment, and let's increase or decrease the amount of that input by plus or minus 20%, and then let's see what happens to the total annual global warming potential in kilograms of CO2 equivalent per kilogram of protein. And as we can see, the buffer addition, when we decreased it by just 20% in value, that decreased our overall annual global warming potential of the entire process by almost 1,000 kilograms of CO2 equivalent per kilogram of protein. And basically what that means is this is a good fix. We can just focus in on this buffer addition for future work and try to either replace it or make it more environmentally friendly so that we'll have more of an incentive to even use these proteins because it won't be impacting our environment in a negative way. And that is basically what the future work will be so that we can help have the incentive to actually use these proteins, sell these protein co-products, help decrease the price of the microalgae biofuels, which will make them cost competitive with petroleum. So we'll start using the microalgae biofuel more and decrease those greenhouse gas emissions. Thank you so much. I'd like to thank Dr. Quinn, Colorado State University. And yeah, thank you so much. That's my project.